Good day, everyone. Welcome to Module 1 of VMCB60 in Virology, where we will be talking about the morphology of a virus particle. Let's start. If you remember this image that I showed last lecture, which shows the different microscopy techniques, electron microscope, the light microscope, and the different organisms and molecules that can be visualized by each technique, you can see that the virus is seen by the electron microscope. All right, so before the electron microscope was discovered, scientists do not know the appearance of these viruses, right? So let's discuss the different components that make up a virus particle. As we have discussed, viruses are very small infectious particles. Some have regarded it as submicroscopic because they can only be visualized by the electron microscope. They are made up of either a DNA or an RNA genome inside a protein shell or a protein coat, and it may or may not have an external membrane envelope. So just with that, we have identified the components, but let's get into the details. All right, let's start with the nucleic acid. All right, the nucleic acid carries the viral genome of the virus. This viral genome or genetic sequence encodes for proteins that the virus needs for its goal of infection and replication. And these proteins are not present in the host cells. So that means even if the virus does not carry a protein as is that it needs, or even if the cell that it infects does not have the protein that the virus needs, it can make that protein since the gene encoding for that is in its genome, all right? And viruses can have either a DNA or an RNA genome, but never both, right? The capsid, or what we call the protein coat or shell, is made up of subunits called capsomeres, right? So in this image, the green structure, all right, would be the entire capsid, and each circle there is a capsomere. And every virus would have a distinct appearance of this capsid. And lastly, a virus can have a viral envelope or not. Now, this envelope is a phospholipid membrane, partially derived from the host cell. So let's get into the details of each component, right? The nucleic acid, as seen here, and I've said, will encode for the proteins the virus needs. It can be DNA or RNA. It can be single-stranded or double-stranded. What we have in our bodies, of course, is the classic DNA double helix. And the nucleic acid can also be arranged in a linear arrangement or a circular manner. The capsid, or the protein coat, is made up of capsomeres, and when we regard a capsid and a viral genome within it, we call that a nucleocapsid. Remember this term because we will be using this as we go along our lecture for this video and the next video as well. All right. Now, what is the function of the capsid? Aside from being the home of the genome, with it, it protects the genome. Remember, this is the most important part of a virus. All right. It makes sure that the genome is delivered to the site of replication, and it also serves various interactions, some various interactions with the different parts of the cell, starting with a cell membrane, with the transport network inside a cell, before replication and when the virus is assembled to be released from the cell. And the capsid can be classified based on structure, meaning yung itsura niya. It could be icosahedral, helical, or complex. It is also called prolate. So let's see what they look like, right? An icosahedral shape of the capsid is composed of equilateral triangles, as you can see there, and they are fused together to form a spherical shape. This is the most common shape of viruses because of the big surface area it has inside to contain the genome. Another shape of the capsid would be helical, or what they call filamentous. This one is arranged in rows to form this disc-like shape, and which forms a central cavity where the genome is located. An example of a virus for this would be the tobacco mosaic virus, which we discussed in the previous lecture, which started the whole study of virology. Going back to icosahedral, there's a lot of viruses that have this shape. The polio virus, the adenovirus, the rhinovirus, for example. And the complex shape of a capsid, or prolate, or the head-tail morphology, is a combination of the two. As you can see, the head is an icosahedral shape. The tail would be a helical or filamentous shape of the capsid. And this is classic morphology for bacteriophages. All right. So another example of the different shapes would be the Ebola virus particle, which is of a filamentous shape. The chikungunya virus particle is an icosahedral shape. So as we go discuss the different viruses, you will be able to see the differences in capsid shapes and how does this differences in shape become significant in terms of the virulence of a certain virus, in terms of how a virus replicates, and how important it is for this capsid to retain this shape once the virus is being assembled. The envelope, which is an optional thing, consists of a phospholipid bilayer and proteins, which are encoded by the virus. And this encloses the nucleocapsid. In the image, this is the pink circular thing. It is partially derived from the host cellular membrane when the virion is released during a process called budding, which we will be discussing in our next lecture today. Now, not all viruses are enveloped, right? We call those viruses naked. And if a virus contains an envelope, it contains glycoprotein spikes or knobs, or what we call peplomeres as well. Now, what is the function of these glycoprotein spikes, right? They serve as the viral attachment to the host cell membrane. They release the nucleocapsid to the cell cytoplasm. They help with that. Some of these glycoproteins have hemagglutinating abilities. Some would have enzymatic abilities that help destroy the cell membrane, and they have antigenic properties as well, meaning a lot of the drugs that we see now that are antivirals target these glycoproteins, which help neutralize the virus and prevent it from attaching to a host cell. Now, they said that envelopes, right, viral envelope, it may serve as an extra protection, yes, but it has a big disadvantage. It is susceptible to the action of a lot of substances, light, we call this the disinfectants and such. We'll discuss this in detail for every virus. So it was believed, and it is still applicable right now, that envelope viruses are more susceptible to those disinfectants, to sunlight, to acidic pH, to extreme environments as compared to the more stable, more resistant nucleocapsid lang. Okay? An example of the glycoproteins and how they are very important for a virus would be the SARS coronavirus 2 particle. All right. So you see there the envelope is the gray covering. You will not be able to see the genome there, but you see different proteins on the envelope, right? You have the red, the big spike proteins. You have the yellowish green, and the oranges. So what are those? All right. As you can see, the spike glycoprotein, that's the red one. The yellow would be the E protein, and the orange would be the membrane protein. And each one would have a specialized list of functions that it serves for the virus to be able to replicate inside a host cell. Another example would be the influenza virus, which contains these red and blue glycoprotein spikes, right? These are the hemagglutinin and neuraminidase proteins, and they play a very important role in the infectivity of the entire virion. All right. If you watch the news or if you are in touch of the different influenza viruses that you hear, you always hear H1N1, H2N3, H1N7, right? Those that name or that code for a specific influenza strain refers to the different hemagglutinin and neuraminidase proteins that makes each influenza particle distinct from each other. That's why you have a lot of different strains and variants. You have H1N1, H1N7, you have H1N5. So these proteins determine the subtypes of the different influenza virus. So that is it for a brief viral morphology. Quite fast. We will go straight to viral taxonomy. Viral taxonomy. It is very important for us to be able to designate names and find a way to classify viruses in an easy way. Number one, to easily study them. Number two, to find a way to see similarities and differences between the viruses in terms of how they infect, how they look, and how does their morphology affect their infectivity, right? But before, we did not have the electron microscope, but we had viruses already. As I discussed in the historical development of virology, a lot of scientists discovered a lot of viruses before the electron microscope was invented, and they were able to name that. But what did they base it on, if not for morphology? They based it on a number of factors, and the most common, and which we still use today, would be naming a virus depending on the clinical and pathogenic properties of the disease that the virus causes. And since this is quite accurate and makes it easy for us to know a lot of things already about the virus without going into the details, it's stuck. This mode of naming viruses still remains right now. An example of that would be the Mureen leukemia virus, which causes, as you may think, leukemia in what species of animal? What is murine? Rodents. Don't Google it anymore. Chicken anemia virus, which is a circovirus, and canine hepatitis virus, right? So you have an idea already as to the disease, which made it stuck, right? Another way that they used to name viruses would be the body sites where the virus was isolated. An example of that would be the rhinovirus, which was isolated initially from the nose, and the adenovirus, which was isolated from human adenoids. Adenoids is a patch of cells adjacent to your tonsils, which is also a member of the lymphatic system. So the tonsils, as you may know, is a parenchymatous organ. You could actually see it, you could touch it and such, but the adenoids is just a patch there on your throat. All right. Another way of naming things, which was classically applied to a lot of things until now, would be based on the scientists who discovered the virus, right? An example of that would be the Epstein-Barr virus, which was named after Michael Epstein and Epstein-Barr. This was the cause of infectious mononucleosis in humans, commonly called mono. They said, I've watched a lot of TV series which showed a reference to mono, wherein they just kiss someone and then they get sick too, something like that. So it's still here, I guess, because I'm not sure if they discovered treatment for it, but it causes mild flu symptoms. Another way to name viruses before, and I'm going to talk about its application now, would be based on the place where the disease was initially observed. So some examples: norovirus, named for Norwalk, Ohio; the Ebola virus disease, named for the Ebola River in the Democratic Republic of Congo; Zika virus, from the Zika Valley, the villages lining the valley in Central Africa. What about the Spanish flu, though? Did it originate from Spain? Sa ngayon na naririnig nyo about sa COVID-19 and how it was compared with the Spanish flu pandemic of 1918, did you do some curiosity-driven work of researching as to why is it that this flu was named Spanish flu? Because from my observation of being stuck at home and watching this pandemic play out, there is some politicians and some people, even doctors, who refer to the SARS-CoV-2 as the Chinese virus, because it was reported first in China, right? In Wuhan, China. However, when you look at and they said, it's okay, look at the Spanish flu, it originated from Spain. The thing is, it didn't, though. The Spanish flu did not originate in Spain. Back then, major countries were involved in World War I, right? And they wanted to avoid panic. So when they saw this, signs of outbreak of a flu in their countries, they suppressed those reports, because that could be seen as a weakness in terms of a wartime censorship. You don't want the ally country to think that the Axis country is becoming weak because they have an outbreak there. So they remain quiet and censored reports. However, back then in World War I, Spain was neutral, and they remained neutral until the end of it. So when they reported of this disease, of this virus, that created a false impression that Spain bore the brunt of the outbreak. So it was named Spanish flu, but did not originate from Spain. Until now, there are debates happening that it could probably be coming from, it came from US, it came from Europe, probably in China, or even in Africa, right? So language evolves, as we evolve, the language evolves. So what may have been an appropriate term in the past doesn't make it appropriate now, right? And even with recently, the MERS virus, Middle East respiratory syndrome, because it was first observed in the Middle East. Now, maybe some people would see that, so that in terms of quarantine, in terms of planning your travels, don't go there. But the thing is, there are a lot of consequences of that, of that kind of vernacular in terms of the people, which we see right now. For example, okay, let me give you an example of a double standard, okay? The swine flu, which happened in the 2009, I'm not sure, sorry. The swine flu that happened then, all right? It was called swine flu. Everyone was using swine flu. But it originated in North America. No one named it North American flu, right? I'm not what do you call this? I am not manifesting like a hate, but I'm observing this double standard. People, Western people always name things, like that. Chinese flu, Asian something. But when it started from them, let's keep it generic, Let's not make it, about us, It's the double standard that I hate. So some people might claim, Chinese virus, Chinese flu is just as accurate as Spanish flu, but it's not, The term for the virus is SARS-CoV-2. The name of the disease is COVID-19, right? So, I won't apologize for touching on that because that's just how it is, That's the truth, and I needed to set that straight. Lastly, some viruses were named based on how they were thought to be transmitted. And when you go into epidemiology, you will see that there's a lot of theories before the cell theory arose, before Koch's postulates were postulated, that there are a lot of different causes or theories about bakit nagkakasakit ang tao. Alright? And some viruses that we see now were named because of that. For example, influenza. It was named because it was the Spanish name for influenced, meaning you were influenced by bad air, because they thought back then diseases, respiratory diseases specifically, are from inhaling what they call bad air or dirty air, or what you call now as miasma. Right, which could arose from dirty organic matter, rotting, those in the landfills, in places where sanitation is bad and such. Dengue is it means evil spirit, because the clinical manifestations of dengue, especially the severe cases, resemble severe fever, chills. Some would have seizures, so they thought this person is, it needs an exorcism or something, diba? So we evolve. We figure out what the people of old have done wrong, and we correct. That's where growth comes from, right? So that's how we name things. Another way to classify viruses be based on the route of transmission, right? Example would be enteric virus, which would be the viruses acquired by ingestion and replicate in the intestinal tract, specifically the enterocytes. Respiratory virus, which would be acquired by inhalation and they replicate in the pneumocytes. These are examples of the viruses. Arbovirus is named for being arthropod-borne viruses, diba? They infect and replicate in insects, and they are transmitted by these insects, which serve as vectors. Right? Perfect na ng delivery, wala pa. Lastly, oncogenic viruses, which actually cause, the machinery of the cells. They target specific cells, wherein they stay there, they transform the function of the cells, which could progress to malignancy, and these are usually the ones causing growths and cancers and such. Now this one, it's not that it's not commonly used, but a lot of viruses right now have what we call large organ tropism, meaning it can infect different organs belonging to different body systems. Right? That's why you have viruses na yung clinical signs niya hindi focused sa isang body system lang. Alright? So, a lot of viruses were not included in this kind of classification because they are overlapping. That is why the Committee in Microbiology established the International Committee on Taxonomy on Viruses, right? They are in charge of developing, refining, and maintaining a universal virus taxonomy classification criteria. All right? They have a website for it. You could see all the viruses and the updated taxonomy for them. All right? Animal viruses, plant viruses, bacteriophages. And it's quite It's quite a handful, especially when we talk about animal viruses. But in animal viruses, there are 7 orders, 96 families, 350 orders, and they're based on a lot of things that concerns the virion itself. All right? The nucleic acid nature, the method of replication, and such. So let's discuss the different levels, all right, hierarchies of the virus orders. So we always start with order, right? That is the highest order in the viral taxonomy, and nomenclature for that, it ends with virales, right? The family, which are based on those characteristics, ends with viridae, and most often, than not, some families would still have variances or differences within its members. So there are subfamilies, and that is suffixed with virinae, right? The genera, which is basically they differ in the viral genome itself. It's called a virus. Some species, there would have a species based on these information. Then you have this types and subtypes, which are based on serological differences, and the strains and variants would be the differences in the molecular characteristic of the nucleic acid. That's specific. So let's, for example, let's start with a common virus, the canine distemper virus, right? The canine distemper virus is the species, okay? And the genera for this virus is a Morbillivirus. The Morbillivirus belongs to the subfamily Paramyxovirinae, which, so easy, the family is Paramyxoviridae, right? And Paramyxoviridae is one of the 4 families which make up the order Mononega viralis. Now, what then are strains and variants? We always hear it right now, or at least I do. I always hear right now UK variant, South African variant, right? Do you as well? We listen to the news. I know we're all tired of the news already, but there's still some benefits of listening to it. So what is the difference between a strain and a variant? All right? Viruses, when they replicate, especially RNA viruses, when they replicate, it's not perfect, Sometimes you don't get the exact copy or the exact replica of the parent virus, all right? Now, some errors can happen in the viral RNA, and these errors are what we call mutations. And new viruses that are produced by the host cell, which are replicated with those mutations, are called variants. All right? It doesn't matter what mutated. It doesn't matter what exact genome change happened. Adenine could be changed with guanine, or all that shitty things. I hate DNA, RNA things. I'm more of a clinical person. I'm medicine. So those are variants. Ano naman yung strain? Basically, a strain is a variant, okay? A strain is a mutated virus in which the mutation caused the virus to behave differently. All right? Si variant, molecular lang yung difference niya from the parent. The strain, may molecular difference, and that molecular difference caused this new virus, this new strain, to act differently. How so? Ano yung mga changes? It could be a change in the cell receptor. It could be a change in the glycoprotein expressed in the envelope. It could be a change in strength of binding during attachment, right? There could be a change in the rate of replication. Mas bumilis ba siya mag-replicate o mas bumagal, right? There could be a change in mode of transmission. There could also be a change in the cell it targets. So these small molecular mutations can ultimately affect the entire study of the virus that you're originally studying. That's why you hear a lot of UK variant, which actually they should name it a UK strain, but they're not wrong when they say it's UK variant. It is. South African variant, Brazilian. I think there's another one of that. And see, since it's hard to name it, based on the genomic sequence, you say it's the thymine and the adenine and guanine. That's why some scientists still stick with the place, with naming it based on the place. They say UK variant, Brazilian variant, So again, it depends. All right. So one big major way of classifying viruses, and you divide them, equally, very clearly with this, is based on the nucleic acid. All right. These are the families of DNA viruses, meaning the nucleic acid of these viruses is made up of DNA, and that DNA could have a lot of characteristics within it. The DNA can be single-stranded or double-stranded. That strand could be positive or negative sense. These viruses could have could be naked, or it could be enveloped. It could also be subdivided into if it is having an icosahedral capsid or a complex capsid. So this is why you have the reviewer assignment for you before the second and the third lecture exam, because you would need to remember each of these characteristics in a summarized, clear, organized way. All right, so you have to find a way to make a table out of this para mas matandaan niyo siya. Because if you think DNA viruses are a handful, just look at the RNA viruses. You have a lot more families to study, right? And the thing is, since every virus would have its own viral characteristic, every virus would have, you would know if it's enveloped or naked, if it's double-stranded, single-stranded. One way to classify all these viruses and make it an advantage for you is using a classification system based on the viral nucleic acid, the strandedness, the sense, and the manner of mRNA synthesis. Remember, ang goal ng virus ay mag-replicate. Okay? It wants to make more of itself to infect the host. All right? You want to make more variant particles to infect more cells. All right? So the process of replication, which we will be discussing in the next lecture, the process of replication differs per virus depending on the the raw product that you have, okay, which is those nucleic acid. This is what we call the Baltimore Classification, proposed in 1971 by virologist David Baltimore, and he divided the viruses depending on their nucleic acid and strands and sense, and came up with 7. Originally, there's 6 only, but then the Hepatnoviruses showed, a different behavior as compared to other double-stranded RNA. So they established a 7th group. We will be discussing this one by one, but I wanted you to understand, okay? The final product that you want is the messenger RNA, mRNA, okay? Which can be read by the host ribosomes, which can then translate the mRNA into the viral proteins, all right? And each virus, whatever nucleic acid it could have, dsDNA, ssDNA, negative ssRNA, it should lead, its replication to mRNA. And when we say mRNA, we're talking about a single-stranded, positive sense messenger RNA, okay? Plus ssMRNA. That's your end product. That's what you have to remember. And you have to remember how they get from for example, class 1, from a dsDNA, how they become an mRNA. How does a class 5, negative ssRNA, become positive. All right? Because the simple knowledge of knowing the nature of the genome, you can think already how the replication happens. Why is it important? You don't have to remember every replication process for every virus. You just have to remember 7. And you have to remember what virus falls into what class. Sa dami ng virus na aralin natin, okay? And each of them have their own unique characteristic. And sa magsasama-sama na sila, you have to find a way to make it easy. And Baltimore, showed us this, which is pretty cool, diba? You just have to remember. Now, here's the challenge for it, which was a challenge I took as well when I was making this PowerPoint, was you have to remember. If you have to review. You have to remember how DNA replication happens, because that's the core, okay? That is Zoo 101, I believe, right? Because it will be hard for you to understand how these work, okay? How this process, the replication works, if medyo malabo na yung role ng DNA polymerase, DNA helicase, Tata box, primers, 5' to 3', 3' to 5'. It will be hard if you're not familiar anymore, at least with those words, okay? So review on that. I reviewed myself as well, okay? And it was good, it's good to review yourself with these things, especially since you're still students. And again, whatever you study before this class or whatever future class you're gonna get or you're gonna take, you are going to need all that knowledge from your past subjects to your subjects now, even if you like it or not. All right, let's start with class 1. With class 1, you start with a double-stranded DNA viral genome, right? This is basically what we have. What we have is, I used the GIF for this. What we have in our bodies, in our eukaryotic cells, would be the DNA double helix. So we know that this DNA genome, the double-stranded one, can directly be transcribed into an mRNA with the help of the RNA polymerase. What's important with this part is the RNA polymerase that the virus uses is the cellular one, meaning it's the RNA polymerase in the cell. Okay? Examples of these viruses that you in class 1 would be the adenoviruses, the pox viruses, and the herpes viruses. All right? Class 2, I'm not going to go into detail as to how that happens, huh? You start with a Tata box. It looks for its primer. Well, that's the primer. The RNA polymerase comes in, breaks up the double helix, breaks up the different strands. I'm not going to go into detail with that. Okay? Review it. Review. What matters is what happens per class. Now, class 2, we have a single-stranded DNA genome, and that could be a positive sense or a negative sense. It depends. The thing is, you have to convert that single-stranded DNA into a double-stranded first before getting transcribed into an mRNA. Why? Remember, your RNA polymerase only transcribes in one direction, that is downstream, meaning 3' to 5'. The thing is, a positive sense is a 5' to 3'. So it needs to be transcribed first. Remember, this is why I asked you to remember the basic DNA transcription. Okay, I cannot anymore. I have a lot to discuss. Class 3, you have a dsRNA, a double-stranded RNA, and this one can easily be transcribed into an mRNA, same as the double-stranded DNA, but using a different enzyme, that is the RNA-dependent RNA polymerase, which is carried by the virus, meaning the virus carries its own enzyme, okay? Not like class 1. Example would be that. Class 4 would be single-stranded positive sense RNA. And as you can see, with what it has already, diba? Ang goal natin is to make mRNA, which is single-stranded positive sense RNA, which it already is. So it can be used directly as an mRNA. It could be translated directly by the proteins, or remember, you're making viruses. Ano pa yung ibang components? You have to make proteins, you have to make genome. So you can convert, not you, the cell. The cell can convert this positive sense RNA into a negative sense first, which is what we call an intermediate, and further converted into a positive sense to make more. And that makes use of the same viral RNA polymerase. Okay? Class 5. Class 5 is the only class which carries a negative sense. Okay? Remember that. Class 5, negative. Okay? And since they only need to be converted into a positive sense mRNA, which can be easily done with the same polymerase. Okay? Class 6. Alright, here is where it gets even more fun. Okay? The ssRNA viral genome is different from the other class because it carries a different enzyme, the reverse transcriptase enzyme. Alright? So what happens in this class? The raw product, which is an ssRNA, is synthesized back by the reverse transcriptase, kaya siya reverse, into an ssDNA. Okay? Just by that. Okay? The normal setting is that the DNA becomes RNA, correct? Ba? But with the reverse transcriptase, you can convert an RNA into a DNA strand. This is then joined by an intermediate strand, which is created with the single-stranded DNA to form a provirus, or the double-stranded DNA. Okay? This DNA is then inserted into the DNA of the host cell, where it is transcribed into mRNA. Okay? So there is this, what do you call this? This reverse transcription that happens with this class. And the classic example for this is a retrovirus, would be the human immunodeficiency virus. This is why there is a lot of opportunity for this virus to stay dormant within the cell, because you are inserting the DNA, the DNA of the virus into the DNA of the host cell, where it can continue to replicate or it can remain dormant. Right? Okay. And it will only be triggered to replicate by a variety of factors. Okay. The class 7 is made up of a double-stranded DNA viral genome, which carries reverse transcriptase as well. Now, example of this is a Hepatnovirus. The double-stranded DNA is morphologically, it is circular and it is gapped, meaning it has spaces between it. All right. And the spaces between it, these segments, all right, what we call segmented, these segments contain RNA as a primer. Okay? And some of these single-stranded RNA primer is used to generate an RNA, counterpart, intermediate. Okay? That is what we call the ssRNA. Now, this RNA is what we could use for the viral protein synthesis. Diba? That's the end game. That's what we want. It's the end game. But that doesn't end there. Remember, we have to make genome. How can we make the RNA into DNA again? That's the role of the reverse transcriptase this virus carries. The ssRNA made from the dsDNA is reversed, transcribed into a single-stranded DNA, and is met with another intermediate made from itself to create the double-stranded DNA that is the original genome for this class. Okay? And then again, it would continue. The double-stranded DNA could make more positive sense single-stranded mRNA. Okay? Oh, I skipped through the thing. Sorry. Example of this, yes, the Hepatnovirus, which is a hepatitis B virus. So, It might seem so heavy, all right? But if you remember, if you just stick to what enzymes to remember and what is the raw product and the end product and what happens in the middle, you got the Baltimore classification dead on. All right, this is just a summary of the Baltimore classification to make it easier for you. That is it for viral morphology and taxonomy. We are going straight into a viral replication in the next video. So thank you for listening to me for the first 45 hours of our lecture. If you have any questions, once again, do not hesitate to message me about it, and I will answer as best and as fast as I can. Thank you, everyone. See you in our next lecture video.